In any case, Sophion, as I briefly touched on this morning, investing a lot in ion channel communities. And when we have these events, we often get asked, what's happening with Sophion? So a lot of things are happening with Sophion. The last year alone, we grew significantly. The interest for ion channels, the interest for our technology is growing, which is a good thing for all of us. And that has led to an increasing demand of hiring more experts within the field, but also to strengthen our front-end operations, to strengthen our whole backbone of what we do. So we're still a small Danish bioscience company, but we have onboarded around 25 professionals the last year. So we're now about 115, and we're continuing to grow. We want to add more resource to our company and more resource to invest in ion channel and in ion channel technology. And when you do that, you also need to move to new facilities because you need bigger labs, you need better production, and you need more space for R&D engineers and scientists. So recently we moved to a bigger facility, still near Copenhagen. So we're still basically only 500-700 meters from where we were the last 24 years actually. We have better accommodations. We do get a lot of visits to our headquarters. So if you want to go and see how a company like us operates, how we collaborate with scientists across the globe, we invite you to come. We have a great facility, and you're more than welcome. And you know the technology is ready and available there. So we do see a lot of scientists actually visiting our office in Denmark. Denmark is also a very, very nice place to have a cold beer, by the way. You've been there, right? And oven sandwiches. And then what we are doing also is that we're building in close to the ion channel communities, close to where you guys are. So that means we have a much bigger headquarter here in the US, based just outside Bedford, outside Boston. Where we have our technology present. We also invite you to come there. We just opened a new facility in Japan. We're scaling in China to see what we can do there and in rest of Asia. So at the end of it, we're a very international company, and we like to be close to ion channels communities around the world. We also spend a lot of time, money, energy on collaborating across the ion channel community. And just in the last 12 months, we've been involved in around 10 scientific collaborations. We hosted 3 of these ICMSs, and we contributed to more than 85 publications. We attended 90 conferences. We had research projects coming and hosting them here. We awarded travel grants. I think we will going to give one here as well. We're also speaking with a lot of leading institutions about instrument grants, etc. So being a private funded company, we like to invest a lot back into the ion channel community. And we're doing that also by being very close to you guys out there. It's a big part of our DNA, as I said this morning. And it's our pleasure to do it. In the States. In any case, we are investing a lot in R&D biology projects. We're investing a lot in bringing collaborations close to the heart of how we develop our technology. And we do that by enabling collaborations closely with scientists across the globe. And we also do it by having more than 20 scientists in our business today. And many of them are working in the laboratories here. We just recently actually welcomed Patrick, who was an employee here at Harvard, to help us facilitate research. And Daniel is going to show you more on that right now and talk about some of the exciting things that we are doing and how we are collaborating with the science community on novel topics. Welcome to Daniel.
Thank you. Thanks. Yeah, first of all, it's a true honor to follow so many outstanding researchers in the field, and I'm particularly excited how many of you are already utilizing automated patch clamp systems in their research and driving it forward. So In the presentation that I have, that I try to really keep short because you do all look very thirsty and I don't want to sort of stand in the way of that. Just want to quickly touch on, now I jumped over the first slide as well, the mission that we have, and Michel touched on it already. So our mission really is to support the ion channel community to do it in different ways by developing technology, tools that you can use to run those experiments and advance your science, but also in our biological group here to develop novel applications that can be used with those tools and really drive it forward in that way. And I have a few examples here that I want to go over that illustrate some of the work that we do here. So just very briefly about the way we interface with the ion channel research community here. I mean, probably not surprising, we're a for-profit company here. We are selling the technology and support our clients in developing their assays and really make sure that they get the most out of that technology. But when we move forward here, more into the middle space here, we also join strategic partnerships with academic or industry partners to develop new assays for challenging targets that others maybe don't want to take up. And that really happens here in that area here. I also do want to point out that we have several grants available that we give out directly to researchers. That could be a travel grant if a grad student want to attend an ion channel conference or a meeting like this one here, but also direct access to our technology where we invite researchers to submit a project and we then have you come out to our laboratory and run together with our scientists these kind of experiments. And lastly, this year for the first time, we started an instrumentation grant that has actually just closed, so no more application this year, but we will soon have a winner for this, where we position one of our systems in a laboratory to really help advance the ion channel research there. All right, so I think everyone knows by now that when these automated patch clamp systems were developed, you can use them for recombinant cells. I mean, we have plethora of different examples available where you show that you can record ionic or you can record currents or voltages from different recombinant cells, but really there has been a push in the past decade to move toward more physiologically relevant assays using more native cells, such as, for example, primary cells or iPSC-derived cells. So for a couple of years now already, we have a neuron project in the lab where we joined different partnerships here. A few are mentioned over there that provided us with different cells, primary cells, iPSC-derived cells with different subtypes, motor neurons, sensory neurons, and many more. And we developed an assay on our automated patch clamp system. So now that's a key challenge. If you want to put these cells on an automated patch clamp system, there's a requirement to have a single cell suspension. And that should be pure. You don't want to have a lot of debris floating around. And that's exactly the challenge to overcome if your 2D culture looks something like that. And you want it to look something like that. So we have invested tremendous efforts into developing novel dissociation techniques, purification techniques to come up with that single cell suspension to put it then on the system. And this has been quite successful. And I want to illustrate that just in one example here in a partnership with Fujifilm CDI, where we were provided with iPSC-derived excitatory neurons and cells that were derived from a frontotemporal dementia patient. And we used those cells, put them on our QPatch system here, and then once you do it, you get a pattern like that you can automatically weed out using some filter settings to objectively only select those cells that really fulfill all the criteria that you want to look at. Once in the recording, you can then go through different steps doing voltage clamp characterization, action potential recording, and even put some ligands on. You can see that being illustrated over here. So one of the key advantages of automated patch clamp is that you really are able to get a snapshot in time. You're not sequentially recording cells on different days. You take up your cells on one day and you can compare disease or patient line toward a control line and have that time-matched control and get all that data within an hour or within 2 hours. And we've done that here, looking at peak sodium currents, comparing the cells that were derived from a healthy donor and those that were derived from a frontotemporal dementia patient, and then compared just the different time points in culture here. And you could see that there's a clear difference sort of separating the two from each other. But this can be taken, you can take this further and look at the concerted action of all the ion channels by recording action potentials. You can see an example of that over here. When we then step to different holding currents and detect the peaks that are being evoked here, we can generate such plots here. And again, when comparing the patient with the wild type, you see a clear hyper-excitable pattern in the FTD patient-derived cells. And that's really in line with clinical representations, but also with what we see in MEA and other assays. All right, so cardiomyocytes have, of course, also been of great interest, and we've done similar studies with iPSC-derived cardiomyocytes. I mean, they really like to stick together. Many of you might know that. So again, you have to come up with creative ways how to get them in suspension, a single-cell suspension, without really harming the cells, so you have a viable cell suspension available for your recordings. And the team in Denmark that was mostly led by Katla Rosholm, my colleague, has really done a tremendous job into developing standard operating procedures for that. So just one example here, these are cells from the group of Professor Nils Voigt in Göttingen. We recorded these cells here, these cardiomyocytes, on the Q-patch system, and we're able to really, with voltage clamp, get different current components in one experiment, sodium, calcium, but much more difficult actually to pick up is here the Herc current, IKR, and even IK1 currents that we were able to record. Again, we went on to look into the action potentials in these cells, and you can see that these iPSC-derived cells do have some heterogeneity in them, but again, the high throughput, beating it with a high number, really allows you to get enough data out to really extract a meaningful result from it. And when we automatically extract parameters like the threshold, the peak, and the hyperpolarization potential, you can actually see that they cluster much more tightly towards each other. It's the APD90 that is more variable here. So you can go ahead and then do a population analysis of such a cell, where we really look at expression patterns in these cells, and that was done here, looking at these different components. And as expected, it requires almost all the other components here, so before you can see an action potential firing. So we have done a technological development that helps to overcome some of the shortcomings with iPSC-derived recordings. So when you do an iPSC-derived action potential recording, you often see something like that. Way depolarized sodium channels, calcium channels are all in the inactivated state, not able to fire. But with our adaptive current clamp feature, it is possible to record the current that flows over the plasma membrane at a defined voltage, here 90 millivolts, and use that experimentally determined value and feed it back into the current clamp recording to then obtain something that looks much more as expected here. So this is a useful feature to use for current clamp, and we look forward to see many of you using it, hopefully. So with the mission to go to physiological assays, we didn't quite stop in the 2D culture. We joined a partnership with a group in Germany here at the Medical University in Hanover. And the team really these are experts in generating organoids using iPSC-derived cardiomyocytes, and they use these bioreactors here to form these organoids, nurture them, and then further differentiate them. And on one day this summer, the team actually took one of the reactors and drove up to Copenhagen, where they then partnered together with my colleagues in Copenhagen to dissociate these cells and put them on our automated patch clamp systems, and voila, we were able to get nice action potential. Oh, yeah, that was too fast. Action potential recordings. I think you saw it here. In a different project here, you could also get contractility readouts. So now completely shifting gears, another field that we are particularly interested in is light-induced, light-stimulated electrophysiology. I think we heard a couple of very great examples during this meeting already. So a couple of years ago already, we've developed a prototype of the Cube. That's our Cube, our 384 platform. I was going for the name Knight Rider Cube. Marketing didn't sign on to that, but it's called the Opto Cube, and it has 384 LEDs below the recording chamber, allowing to irradiate light during the recording into the chamber. With the QPatch Compact now in the market in many of your laboratories, we of course got the question as well if that can be done with that technology as well. And indeed it is, now it has a life of its own, it is indeed possible to use blue light radiated in here. And this is a nice example where we had a recombinant GABA cell line and we added a caged GABA compound, ruby GABA, into the flow channel, and then irradiated blue light that caused the uncaging. And then you can see this activation of this GABA current here once you do it. So I think there are many applications, and that is just one example. Another one that we've done on the Cube system in partnership with a Zero company in Italy, Axam. So they had their hands on a recombinant cell line expressing HCN2, and it also co-expressed a bacterial photoactivatable adenylate cyclase that converts ATP to cAMP when light is irradiated onto the cell. So cAMP then in a downstream mechanism via pKa leads to phosphorylation of the HCN channel, which then causes activation. And that you can record with patch clamping. So we put that cell on the system and did a step from -30 millivolt to -90 millivolt. And then we obtained here this baseline current. Then we irradiated light for 5 minutes into the chamber, and you see that pronounced activation of the HCN2 current. You can also quantify that by looking at the activation curves here that really causes that right shift, the depolarization shift here when you optically stimulate it. So yeah, please, if you have more ideas for that, please feel free to approach us. We'd always be eager to hear how you can use these tools to drive your research. Size matters. At least we know that's the case if you want to do red blood cell or organelle recordings on an automated patch clamp system. For that matter, we have developed new consumables that have a higher resistance, a smaller hole size, and we've tested it with, in a first step here, using red blood cells. But we also have efforts that I'm going to touch on briefly to record from various organelles directly. So you can already purchase the Cube, that's the 384 consumable, but we will soon have it available for the QPatch and QPatch Compact as well in the beginning of next year. Yeah, so there are different channelopathies in the piezo and the Gardos channel in red blood cells that we are trying to study here in collaboration with Novo Nordisk and a large hospital in Denmark, Rigshospitalet. And so we developed an assay for these red blood cells, and that's of course not easy because they're very small and flexible, elastic as well. But Kian, my colleague in Denmark, was successful developing an assay. And I just want to show this one data set here, where he first used a recombinant line expressing mouse piezo 1, and he applied a voltage ramp and then added this activator compound that caused the increase in the current, and you could inhibit it again with gadolinium. Now looking at the red blood cell, a similar phenomenon was observed here. Adding the activator led to the increase, and you could inhibit it again. And just for the control experiment here, the recombinant GABA line that didn't show that effect. And this is an ongoing collaboration, and we expect to see more data from that in the future. Yeah, so just very briefly, I touched on the lysosomes here. We formed another industry partnership with a French startup company, Auria. They really specialize in high throughput isolation of lysosomes. And together with Sophion Biosciences, we are developing an assay on our automated patch clamp system that allows to record currents from lysosomes directly. So just with my last slide here, I want to touch on a collaboration that is pretty early, but is some of the evolving fields. I mean, obviously, immunology is definitely not a novel evolving field, but I think ion channel targets are just more and more surfacing now, and groups are pursuing them as drug targets. And with the group of Stefan Fesquet at NYU and Sumin Song of the ION Lab, we set out to develop assays for recording currents from primary immune cells, such as T cells or macrophages. And our plan is to host a workshop in our laboratory in Bedford in early 2025. And if that's something that's interesting to you, then please stay tuned on our webpage. We'll be making an announcement about that in the coming months. So now, already over time here, I see some people start looking angry at me. So I want to point out, though, again, our grants that we have available. If you are interested in a travel grant or accessing our technology, find a Sophion night here during a meeting, talk to us, or go to our webpage where we have more information about these different grants. So yeah, with that, I want to say thank you for your attention, for sticking around until the end. And yeah, looking forward to seeing more talks tomorrow then.